Hello students, welcome to ePathshala. I am Dr. Subhradeep Karmakar from the Department of Biochemistry at All India Institute of Medical Sciences, New Delhi. Today we are going to talk on module of gene analysis from the paper Bioinformatics. This module will discuss about the regulation of gene expression and methods to quantitate the gene expression by performing what we call as gene analysis. Now before we go into that, we need to understand a few things and few key concepts so that you can understand the gene analysis in much more appropriate way. The various things are the eukaryotic gene structures, the regulation of the gene expression, and the methods to study gene expression. Now there are different ways to achieve these particular objectives, and today we are going to focus on microarray based gene expression, and therefore we are going to talk a lot about the microarray data analysis pipeline. The gene expression, as you know, is fundamental output of the DNA. So DNA is the nucleotide sequence, but the functional output of the DNA is in terms of the messenger RNA which it produces, which is called the transcription, and that is the functional output of the DNA. So studying gene expression is fundamentally knowing the functionality of a genetic material. So if a gene is very active, then it is going to produce a lot of transcripts. So we say that transcription it is very active. And on the other hand, if a gene is silent, then it's going to produce fewer transcripts, and then we are going to say transcription it is less active or silent. Now there are various methodologies that have been adopted by scientists to study the gene expression, and those are actually been classified into various categories, but we are going to classify them into two major categories, which is known as the low throughput way and another is known as the high throughput way. PCR, as you know, it's a kind of a standard gold standard to measure the gene expression, and it is a wonderful techniques, and it has revolutionized the way in which we kind of study the gene expression. But then PCR has a few pitfalls, and these are not really the pitfalls but kind of shortcomings, that you have just a limit of how many gene transcription you can actually modulate by PCR, because it's a low throughput techniques. On the other hand, the techniques like microarray, and these days we have the next gen sequencing, which are extremely high throughputs, and you can actually interrogate together hundreds and thousands of gene expression profile in a single experiment. Now today we are going to talk about microarray based gene expression profiling, and microarray is very similar to kind of the next gen sequencing, except that there are a few differences which we are going to talk later. But microarray can interrogate thousands of gene expression changes in a single experiment, and thereby provide an important driver in this field. Fundamental to microarray studies is the data analysis pipeline, because once you do the microarray, the next question is that what are you going to do with that data? It's going to produce a large data sets. So the next important thing is that to do the mining of the data or the data analysis, that is often very poorly understood, and this module is going to address a few aspects of this particular of this area. Let's know about this eukaryotic gene structure. So eukaryotic genes compared to that of the bacterial genes is a little different, because bacterial genes doesn't have what you call as the introns, whereas the eukaryotic genes has two different parts. One is the coding regions, which is known as the exons, which is interrupted by the non-coding regions, which is known as the introns. And in addition to that, a gene must have in start sites, stop signals, the regulatory elements. So this cartoon actually show you the typical eukaryotic gene structures. It's kind of differs from gene to gene, but it's kind of an average. This is what you can see in majority of the eukaryotic genes. So there's an upstream enhancer region, which we call as the regulatory regions. Then there is something called the proximal promoter regions. That's the region which actually recruits RNA polymerase II and transcription takes place. And then there are a few more regulatory elements or regulatory boxes, which are the Tata box and GC boxes. And then you could see over there are the red color boxes and the white color boxes. The red one are the exons, which actually are being spliced and joined together, and the white colored one are the introns, which are actually spliced out, and this is through a process of splicing. And then at the distal of that, we could see there could be the stop codons, which actually terminates the process of the reading of this RNA. And then we could also have some enhancers, which is in downstream, which we call as downstream enhancers. Typically, on an average, the eukaryotic gene actually of about 7 to 10 exons, they actually spreads that long, and they had about 10 to 16 kb of DNA. We are going to first talk about the genetic code. We all know that it's a triplet sequence of mRNA that actually specify amino acids. Now with 4 different nucleotides and a triplet codon, the maximum size of the codon is going to be 4 raised to the power 3, that is we have 64 codons. Now 64 different combination of the bases 61 of them actually codes for amino acids, and the last 3 codons we know these are the stop codons, and they really don't code for any amino acids. We call as one termination codon. And another very important thing about genetic code is that they are known as the degenerates. What does that mean? That means that more than one triplet codon actually specify the same amino acids. Say for instance, the amino acid serine is eventually encoded by 6 different codons. Now there is something called unambiguous. So each genetic code actually specifies a particular amino acids. The codon like TGG codes for the amino acid tryptophan. Now there is something called the non-overlapping. This means that successive triplets actually been read in an order. So each nucleotide is actually been part of only one triplet codon, and this non-overlapping is very important because if there is a slip of a single codon, then it's going to read the next codon, and therefore that is going to be an issue or problem. Exploring the eukaryotic gene structure. This is a little continuation of what we had seen in the previous slides. You could see the exons which is actually been interrupted or punctuated by the introns. And then is something called the splicing process that takes place, and that's a very interesting phenomenon where there is the different shuffling of the exons that actually takes place, and depends upon which kind of exons or which exons actually been joined together and which exon is actually left behind. We have the different transcript variants or different isoforms of this particular gene, and that actually gives a vast defined repertoire of a particular gene into different RNA. So you could have one fundamental gene, but at the same time, because of this alternative splicing and then keeping a few exons and then actually leaving aside a few more exons, you could have the different splice variants or the different isoforms, and that's very interesting. It's going to increase the entire functional output of the genes of a particular gene several fold time. And we had actually seen this example a lot in the case of antibody select. We had seen them several times during the process when the antibody genes actually been shuffled in that way. Now we are going to talk about exons and coding sequences. So what is the differences between exons and coding sequences? Well, often exons are actually defined as the region that actually defines or that actually produces proteins or amino acids. Now that particular definition is a little outdated because we are going to refine that definition in a little while just now. So exons are often defined as short segments of the protein coding sequences. True, but this is a bit of oversimplification in this, and it's not always true. So exons are actually the segments of sequence that actually been spliced together after the introns actually been spliced out from the pre-mRNA to form the mature mRNA. Even then, not all the exons actually encodes to the protein, and that's the thing which we need to remember very strongly. There are certain exons which really are the part of the mature RNA, but still they doesn't translate to any proteins or amino acids, and call them non-translating exons. Majority of these non-translating exons actually forms a part of what we call the untranslated regions or UTRs. You may have heard of this. There is the two UTRs. One is the 5' and another is known as one is another at the 3'. We call as 5' UTRs and 3' UTRs. And most of the time these UTRs are actually been made of the non-translated exons. The UTRs actually been found upstream and downstream, as I was talking just now, of the protein coding sequences. So in summary, many exons just codes for the UTRs and not for any amino acids or proteins. Regulation of the eukaryotic gene expression. Now genes can be regulated at many levels. Usually when we speak of the gene regulation, we are actually referring to transcriptional regulation vast majority of the time. Now the complete sets of all the genes being transcribed as we are referred to as a transcriptome or entire transcriptome. Now there are different stages. You could see the cartoon below. It actually shows that the DNA folded on itself, that is the DNA is actually replicating. And there could be a regulation of the gene expression even at this stage, when how frequently the DNA gets replicated. We know that a cancerous DNA gets actually replicated pretty much more number of times than a normal cell. So that's another step of gene expression regulations. And then the DNA is actually transcribed into RNA. It's called the transcription. That's a major control hub where majority of the regulatory process or regulation takes place. And then there is something called the RNA translation into proteins. That is another regulatory point that's known as the translational regulations. In addition to that, or in addition to this, which is actually depicted over there, we have the RNA decay or the half-life of the RNA. That is another point of the regulation. A few RNAs may be degraded very fast, a few may be staying for a long time, and that is also another important point of the regulations. And then we could have the control of how the RNA is actually coming out of these nuclear ports and then actually goes to the ribosomes. So that is another point of the control of the RNA regulations or transcriptome regulations. In addition to that, these days we have a lot of something called the non-coding RNAs, the microRNAs and the long non-coding RNAs that actually somehow go and inhibits either inhibits or somehow activates this particular transcript. So there are different complex ways of regulating a eukaryotic gene, and there are different ways to do that at the different levels. So utility of studying gene expression analysis, or why do you need to study gene expression? Now this studying of the gene expression or transcription is going to give us a snapshot of the RNA expression profile in a cell. Now this has a lot of importance and a lot of utility both in the basic sciences as well as in clinical sciences. Now for instance, cancer diagnosis, drug testings, and pattern of gene expression during developmental processes or during differentiation programs is very important, not just to know what is the gene expression profile at a particular point of time, but rather it is more interesting and more informative to know how the gene expression profile changes over time. So you have an initial stage and you have a later stage, and it will be very interesting for us to understand how this later stage actually evolved from the initial stage. So this is what we call as the dynamic change in the gene expression, and that is what is actually very important for us to understand the biology, and that can show how the RNA level also changes during the development process, like for instance drug treatment or a cell actually transformed from a normal to a cancerous phenotype. So all of this kind of transformation is very dynamic, and these have been associated with a pattern of the gene expression changes. Now the microarray based data analysis is also very important to monitor these particular gene expression changes. And if we could just conceptualize, you understand that when this kind of important biological process takes place, then hundreds and thousands of gene profiles actually are altered, and that is actually the most important thing that we need to do in single experiment, so we can capture all this that has been happening in the dynamic mode of those 25 to 30,000 gene expression profiles during a particular process. Methods to study the gene expression. As I mentioned, there are different methods to study the gene expression, and that's the kind of the timeline which actually shows you that back from 1953 to 2008 to 2016 and 17, you could see there is a huge evolution in the path and dramatic changes in the way in which we study actually the gene expressions. So we started with something called the northern blots and PCRs and the RNA protection assays. I don't say that these are actually obsolete, these are still very well used, but then the wealth of information which you get nowadays using a single experiment is enormous, completely mind-boggling as compared to what you used to do earlier, a couple of decades back. Now, if you are going to integrate a single transcript, there are different methods like northern blots, RNA protection assays, RT-PCRs, real-time PCRs, in situ hybridizations. Real-time PCRs or PCR-based techniques are extremely good, they are gold standards, but then there is a limit of how many number of PCRs you could perform with a particular gene expression profiling. You could do 96 samples, 384 samples, and that's the kind of a limit. But beyond that, if you are going to do a different kind of an arrays, different kind of techniques which are going to talk today, there's a microarray based techniques, we could integrate a large number of changes in the gene expression profiles in one single experiment, and that's amazing. So we are back in 1999, the DNA microarray technology came then, and after that, there is a complete revolution in this particular field. And now we are actually dealing with what we call as the next gen sequencing techniques, which is actual sequencing of the transcripts. And this is an extremely good platform for doing what you call as a de novo discoveries, where you have no prior knowledge of what the transcripts will be, what are the sequences, but then you just sequence them and then actually map it to a reference genome and then find out exactly what are this nature of the transcripts, or what is the identity of these transcripts, and that's very interesting. The methods to study the gene expression. It has become possible to look into the entire transcriptomes in a single experiments. Now back a few years back, even a few decades back, we have a few techniques which could interrogate the change in the gene expression profiling, and they are mostly gene by gene basis. Say for instance PCR, you could profile at the max say 96 samples together, or if you're a little more sophisticated, about 384 samples, but that's the max we could achieve in a single run of the PCR. But what about a scenario where a particular event actually takes place? Say for instance you treated a particular cell with a drug, and then there is a change in the, we want to understand what is the entire change in the transcriptomes. No, then in those scenarios a PCR based profiling of the gene expression is really not the right way to go, because you could just do in a very low throughput a few genes at a time. But then this kind of necessities actually has given rise to two most fundamental methods to study the gene expression changes with a large number of in one single experiments. And the most two common things are the microarray based profiling, another is known as the sequencing based profiling. The microarray is actually been based on the hybridization of the complementary probes with the transcript, and then there is the sequencing based approaches, which is kind of very trendy these days, and vast majority of the data and vast majority of the scientific discoveries are based on sequencing based approaches these days, and they are actually the real sequencing of the transcripts, really sequencing individual transcripts. Now this sequencing based approaches could be sequencing the transcriptome, sequencing the genome known as the DNA-seq or RNA-seq, and they are extremely powerful and they are very informative. But we are going to focus today mostly on the microarray based profiling, and there is a definite reason for that. Number one is that not everyone can actually afford to sequencing based profiling. The reason is that it is computationally very challenging. So if you have in sequencing data, it takes computational power to mine the data and then get the important information out of that. So that's not which every lab can actually do it very easily. And number two is that it is little more expensive, even though the price is dramatically coming down every day, but still it is expensive beyond the reach of many labs. And therefore we are going into another approach, which is microarray based profiling of the gene expression, which is almost equally informative like the sequencing based, but way less expensive, and the data is a little less computationally challenging. So many softwares and algorithms are there to mine the data, and you can easily get the information out of that. So currently the microarray based platforms is actually based upon the probes which are actually been printed on a glass slides, and we are going to hybridize our RNA or the transcriptome, if you are interested about transcriptome, the entire RNA onto those probes, and then individual of these RNAs will be leveled with a specific dyes, and then eventually you are going to read that. We will have a slide following after some time where you could see the entire workflow of these microarray experiments, and then depending upon what color actually comes out from the slides depends like how the hybridization took place with the particular transcript and the probe that has been there on the glass plate. We are going to tell that whether the gene has been actually expressed or it has actually been silenced. Now this is the first high throughput technology for studying the gene expression analysis, and it has been over so many years that even though we understand that this sequencing technology is taking over, but still we are kind of stuck still with the microarray because it's so easy to perform and it is yet so informative. Now if you a more recent technology for the large scale gene expression analysis is actually been used is the RNA-seq information. Now that's actually the sequencing. I was talking a little earlier that we actually take the actual transcripts and then sequence them. So that is extremely important for discovery kind of scenarios where you need to make some de novo discovery, but then that has its own challenges and own shortcomings in the terms that it is computationally very challenging and not everyone can afford to do it in the first go. Now these are the deep sequencing techniques, and they actually gives the RNA output or the RNA expression in the terms of what you call as an FPKM values. So it's kind of a little to be understood in a way that if the FPKM values is higher, this means the transcript expression is more, and if it is lower, then it is actually less. And it is a very reliable matrix to understand the gene expression profiling. That is all in the deep sequencing based technologies. Now why microarray? Now when you talk about microarray, we just have to again understand that it's just not the transcriptome, or it's not just not the RNA expression, but it can actually be used into different scenarios to understand and interrogate the different kind of biological problems. Of course transcriptional profiling is there, but in addition to that we can do the gene copy number, we can do resequencing, genotyping, and there is something called CHIP assays, this chromatin immunoprecipitation assay, where we need to find out or we actually address that what are the transcription factors that actually binds to the DNA. Even in them, there we have this hybridization based systems which is known as chip on chip, where you take the immunoprecipitated DNA. After we immunoprecipitate the transcription factor with an antibody, then you eventually purify the DNA and then you hybridize it on a glass slide. So that's an immunoprecipitation based hybridization techniques, that's chip on chip, but that is going to integrate and answer a different kind of questions. And then there is something called the tiling arrays, where the different probes across the entire length of the genes overlapping with each others with a few nucleotides are actually been layered on the glass slides, and then you can actually ask this specific question, is that where exactly a particular transcription factor or a particular protein is actually been recruited on the DNA. So those are known as the tiling kind of an arrays. So you could see that the entire hybridization based technology can actually be improvised to address different kind of biological questions depending upon what kind of scenarios and what kind of problems you actually been addressing. Now this is the instrumentation portion of doing a microarray experiment. So the first one actually shows you a 16-pin microarray spotter. That's kind of a very sophisticated instrument that actually literally prints using a printing technology those probes on in particular slides, and the probes are the one that is going to hybridize with our RNA transcripts from the samples and is going to give us a particular color, and this is we are going to just see it in a little while. And the next one actually show you a gene chip, the actual gene chip from an AFE matrix. You could see how small it is and how compact it is, and still within that particular chip you can actually integrate hundreds and thousands of gene expression profiles. And the next thing is that you take the samples and you actually label them with the different dyes. So you're going to label them with mostly two different dyes, Psi3 and Psi5. We're going to talk that about in a little while. And then actually put on the particular gene chip and then actually do the reactions, and eventually you read them with the help of what you call as microarray scanner. And once you scan it and read it, then you're going to get bunches of dots like that what you see in the last cartoon, the red and the green and the yellow dots. And that actually shows you, say for instance, the red one is actually the control samples and gene expression profiles in the control samples, the green one is the gene expression profiles in the test samples, and you're going to see the nice pattern of the expression profile changes in a particular slide. And we are going to convert these particular signals into the light into actual quantitative value, and that is what is more important for us, for the biologist, to interpret the data. So what are the steps? So having finalized on the microarray in these particular modules, next the question is that what are the steps for microarray data analysis? So there's different steps. One is, once you have the data, then you need to filter up the data and remove the junk or the things which are not that important for us to proceed further. That's called the data pre-processing. And following that, there is something called the normalizations, where we are going to take care of the fluctuations in the data due to so many different reasons, not because of the actual change in the gene expression, and that's what we call as the normalizations. And then we are going to apply different statistical tool to look into the data, that how rigorous it is, and the experiment, whether it has been performed correctly or not. Now gene analysis, as I mentioned, is that it actually involves a lot of statistical analysis and statistical tools, so that the data what you get out is very rigorous and can be have very high statistical power. Now after you found out a few gene expression profile changes associated with say a particular process, the next important thing is that we need to categorize those gene expression profiles into what we call as the different clusters, which is kind of finding out a pattern within the huge gene expression data which we get from a microarray experiments. That is called the clustering, and we are going to use different clustering algorithms to find out the pattern within the data set. The microarray DNA data analysis pipeline. This is actually an algorithm of the different things which you actually normally do during our microarray experiments. We have in particular experimental questions, and for that we design an appropriate experiments, and then we do the sample preparations, and then buy the chip or an array, and then we hybridize that particular RNA after labeling them with a particular dye on this array. What you get is actually bunches of dots which is show that what you have shown in the earlier slides, and that is what performs the image analysis portion of it. And once you have that, then you do what you call as the normalizations. Now this is an extremely important steps where you kind of take into considerations that during any experiments there will be little variations in pipettings or other errors, and those are the technical variations, those are the variations which has happened because of some technical reasons, not because of actual biological process that took place. And therefore you need to make sure that we kind of normalize that, so that we don't comment on those factors as the actual changing in the gene expression. Rather, these are just the experimental errors or experimental backgrounds. That's very important. And then we do what we call as differential gene expression profiling. And of course, the most important thing is to use proper statistical tool to derive and make this particular data really worth and valuable. And therefore, and there are different modules for that. You could do the microarray experiments and just look into what you can say in the gene expression profiling or the whole transcriptomes. You may not look into the whole transcriptome altogether in one particular array. You might look a part of the transcriptome. So you may have a pathway specific transcriptome changes. Say for instance, if I am interested in seeing that how the T cell gets activated upon stimulation with a particular antigen, so you could take a very customized array and then look into that. not in the entire genome, but only into those portions where there is the T cell dependent pathways actually been focused into. So those kind of customization is always possible in a microarray experiments. What are the different experimental considerations and designing for a microarray experiments? The most important thing is that replicates. Now, whenever we do any experiments, in order to understand that whether really the data is valid or not, we need to repeat the experiment several times, right? And that is what we call as the performing the replicates. There are two types of replicates. One is known as the technical replicates, and other is known as the biological replicates. Technical replicates is essentially doing the experiment one time and then dividing into different parts and then doing the assays. That's fine, that is going to take care of the individual variations in the pipetting and other processes, but more important for us is the biological replicates. Biological replicates is redoing the experiment again and then trying to see the reproducibility of it. Say for instance, I did an experiment over here and using a particular cell, and then I do the same experiment after a couple of days after, say a week after, a month after, in a different locations, using maybe the same cell from a different passage, and then trying to I understand that whether the data which I actually obtained in the first time is reproducible to the data which I am getting now. So that's what is known as the biological replicate. And for a biologist, and to understand the gene expression profiling, the biological replicates has way more significance. Of course, we don't say that technical replicates are not important, they are important, but it's more important to have the biological replicates. And then there are certain steps called the amplification of the RNA. These are a little technicals, like if you need to do the microarray experiments, then many a times it is good that once you have the RNA isolated, good RNA, quality RNA, undegraded RNA, then you are going to amplify the RNA so that a few transcripts which are kind of little expressed or they have a less copy number, we don't miss out those transcripts. So we need to have an amplification step of that. And then something called the dye swaps between the replicates. Now this is we are going to appreciate a little later, but just I am going to tell you that most of the time whenever we have the control samples and the test samples, we are going to level them with the different dyes and then hybridize it on the probes on a microarray. Now once you suppose level the control with the red dye and test with the green dye, then we are going to redo the experiments by doing the alternative way, that control has to have another dye what the test was having and then just swap it. The only reason for that is that we wanted to make sure that the expression profiles what is changing is not because of this dye, but is because of the actual change in the transcript. So this is a way of an internal quality control, and that's a very good practice to do in the microarray experiments. And of course we have something called the reference samples, some kind of a standard which is going to tell us that everything is going on good. So this is what I was talking about, that technical versus biological replicates. So although technical replicates can be useful for reducing the variations due to the pipettings and hybridizations and everything, biological replicates are extremely important, and they form the basic backbone or fundamental reproducibility of the experiments, and that is actually to be never ignored of, and it has to be repeated in at least 3 biological replicates to get in statistical significance. So this is pretty much the same that we're talking about, that the technical replicates are part of the same assay dividing into 3 fractions, whereas biological replicates are totally new experiments which is actually been done in a different setups to see the reproducibility. So this is the workflow of the microarray. The right hand side of the panel actually shows the different steps which we are talking a little while. The sample preparations, isolations of the RNA. Good quality of the RNA is fundamental prerequisite for doing any successful microarray experiment. So the RNA is degraded or is poor quality, avoid doing that, because that's not going to give you the actual results. It's going to have a lot of errors. So please avoid this degraded RNA sample. So one way of checking the RNA quality is something called the RIN score, which is RNA integrity score. And any RNA whose RIN is less than 8 is to be avoided. Any RNA having a RIN of 8 and above up to the maximum of 10 is the pure RNA, good RNA, and that's what we are going to use for the experiments. Also, it is a good practice to take this RNA and then run it over a gel, not just take the 260 to 80 ratios in a nanodrop, but just run it to the gel, and you're going to see 2 main bands, one is the 28S and this 18S ribosomal RNA. Generally, the ratio should be 2:1. The 28S RNA should be 2 times the amount of 18S RNA, and if that ratio looks into a gel, kind of 2.2:1, we're a little confident that this particular RNA is of good quality. But if you see a smear, if you see a lot of hazy in the background, if you see that the ratio has been reversed or not been there, then that gives a first indication that the RNA is compromised, and it's better to avoid that doing experiments using that particular RNA. and after that this RNA is converted to cDNA, and then you're going to label them with the different fluorescence dyes and overnight hybridize them with the probes on the microarray, and the next day you're going to see the hybridization reactions by reading it under what you call as the microarray reader, and then of course do the normalizations and then actually take the data out. Now the left panel actually show you how the gene expression profile actually looks like. Suppose we have three different conditions, 2 and 3, and you could see that certain genes are actually been commonly been altered in both the condition 1, 2 and 3. That's the common sets of the genes, but there are few genes which are very unique in condition 1 and not in condition 2 or condition 3. So those are actually pathway specific or experiment specific alterations, and that is what we call the entire thing is what is called as the differentially expressed genes, and that is what is most important in any kind of microarray based experiments or for our biological processes. So the experimental design for microarray amplification of the RNA, this is something what we are talking a few minutes back. That's an important step when dealing with less amount of sample. Let's take a scenario where you're going to see, suppose a few developmental genes during a developmental process, a differentiation process. Now, we know that majority of the developmental genes are present in very small copy number, or they are a very small representation of the bulk of this RNA or the transcriptome. Now if you are not doing this amplification step, it is very possible that we are going to miss out the signals of those low copy number transcripts, and then you are not going to get the real biology out of it. So if your target genes are those genes which are little expressed or they are low copy number, then it's always a good practice to amplify your RNA so that they are actually being represented well in the population and don't miss them out, and that is actually very important. And there is something called the linear amplification methods can be used to increase RNA so that the microarray experiments can be performed using small number of the cells. Now it's not clear to what degree this affects the results. In fact, if you go through the different papers, then there are a lot of disputes and there are differences in the opinions, with some groups which are in favor of RNA amplifications, whereas some groups says that it doesn't make a huge difference between this. Nevertheless, it's always a good practice to amplify the RNA and proceed with your experiments. So we are talking about dye swaps. As I mentioned earlier, that we have the two samples, and the two samples, the control samples and the test samples, say for instance, they are actually being labeled two different dyes. So the control samples is labeled with the red dye and the test samples is labeled with the green dyes, and then we are going to take these samples and hybridize in an array, and then in the next round we are going to redo the same experiments by swapping the dye. Now the control gets the color while the test was in the previous experiments. This is just rule over and rule out any biases that is because of the different dye that is actually used, and ideally you should see the same results both of the time with the little variations within the acceptable limits. But if you see a wide variation, then that actually concludes that this particular dye is the one that actually plays a very important role in the variations and not the real biological process, and that therefore these dye swap experiments are very important. Most of the time we are actually labeling them with the two different dyes, Psi3 and Psi5, that is actually been used for majority of the microarray experiments. So experimental design for the microarrays, of course we are going to use the reference samples and housekeeping controls. Of course this is very important because we need to make sure that a few genes really doesn't alter. That actually gives some kind of a benchmark for us to understand that there is a background control, and anything which has been altered over that control is what is the real thing. If we see a variation in the controls, then we are really not sure whether we see a variation because of the difference in the amount of the sample we loaded, or because of the actual or the real variations. That actually makes the importance of using references or internal references as an QC or quality control in doing an microarray experiments. So you could see S1, S2, S3 are the three different samples, and the corresponding references are R and R. We are going to do an S1 by R, S2 by R and S3 by R. We are taking the individual samples and going to divide them or going to normalize it with their own references. That kind of forms the benchmark for the normalizations as in control. Now this is the gene expression data plots, called also sometimes the MA plots. So the gene expression level is the ratio of the gene which is actually been expressed in a particular RNA samples or mRNA samples. So we are going to do what we call as the M, which is essentially the log2 fold of the red intensity by the green intensity. Of course red and green are something which we are going to define, whether our control is red or whether our test is red. So this is our convention. And then when you're going to do the ratio of these two, that's the M score, and then we have the average of these two, which gives us the A score, and that's actually the average of the log2 fold intensities of red and green and the average of that. So you could see that this MA plot is kind of a very important plot to know the variation in the gene expressions. And this is the cartoon that actually shows the plot both into the log into the M by A, which is actually plotted in this particular axis. So how to submit a microarray data? So once you have the microarray experiment successfully done, the next important thing is that you're going to possibly mine this data and got a very interesting result out of that, and written a paper and then communicated that to a journal. Then the next thing the journal is going to ask is that you need to submit it to the public domain, so that your raw data, whatever you obtain, can actually be mined by other peoples and other groups, and they could check, number one, whether our analysis is correct or not, and number two is that they might have to do with some other informations which we are not interested in. But there is a definite way, definite syntax in which you need to submit the data, and that is extremely important. And where you are going to submit the data, there are different repositories which actually access the microarray data submissions, like GEO is one of them, where you're going to have the microarray experiments. You're going to put an ID for that particular experiment and submit over there, and anyone can actually pick up that particular ID and then take the actual raw data and reanalyze it in a different way. And that is actually forms a core fundamental principle of data sharing and checking out the reproducibility of our experiments. Now the data can be submitted into different formats, like Excel formats is one of the most common way of submitting the data. So you talk about a term known as Miami, which is minimal information about microarray experiments, and that is very important, that when you do an experiment you need to come up with a few minimum information about how the experiments were done, how many replicates were there, what are the starting materials, what kind of dye did you used, and other things, which totally forms one composite package that actually helps the users and other individuals in the public domain to know a little more about our experiments. Now the experimental design type, factors, types of array, reference samples, and what are the sample used, and how they are actually been labeled, and what kind of dyes we are actually used it, what is the quality of score of our RNA. All these are the things that actually forms a part of that minimal information package. Next is the measurement of the data and specifications, like quantifications, hardware and software used for the scanning, and the raw measurements and everything, and also the type of the microarray. If you see any microarray experiments, there are a few companies that actually, few commercial vendors that actually performs or sells the microarray chips, like Illumina is one of them and Agilent is another of them. So if you have an Agilent data and if you're going to reproduce the same experiment using an Illumina data, you need to have that information beforehand, that you are going to compare the data across the two different platforms, because they will have a little different in the architectures, little different in the nomenclature. So if you don't tell out to the public that this is the kind of things which you have used, this is the vendors, this is the type of the array, then it's very hard for someone to find out exactly what or how we did the experiments. So things to remember in microarray analysis is that microanalysis is complex and is rapidly evolving field. So even though it is kind of started way back and sequencing technology is picking it up, but still it is something which is rapidly evolving, and there are so many different variants out of it. Special care has to be taken to do the normalizations, which is kind of making sure that we are seeing an actual change in the transcript, and that is not because of any experimental errors. And between the arrays and within the arrays, and there is something called the reproducibility and replication of our experiments. So we need to do a couple of biological replicates to make sure that the data what we obtain now is also the data which you are getting it again and again time. And then something called the false positives and false negatives, which are going to talk a little more, and the p-value considerations of this. So although a lot of different software packages have freely been available to do the data analysis, consultation with an experienced statistician is very important, because that person is going to guide you that what kind of tools you are going to use, what kind of analysis you are going to do to make the maximum use of the data. So it is always better to consult and statisticians before you start the experiments, rather than do the experiments and then rush to a statistician for getting his help. That's not the right way to do. Right way is that first take and build a core team where a statistician should be there, and then discuss in length with him that what is your problem, what is the biological problem you are kind of addressing, and then that particular individual will be able to help you and tell you what is the right way to design that particular experiment so that you get the maximum information out of it. Data analysis, what genes are differentially expressed. Early days, the fold change cutoffs 2x the differences or better is what is something what was regarding and was not a very satisfying approach, because of the reason that if we assume that, if we think that anything over twofold is what is important and less than that is less important, is something sometimes an oversimplification, because you could have a twofold change or a 2.5 fold change, but still we could see that that actually makes a lot of differences. So just don't go with the fold change, or it's not something like this that a larger fold change means very interesting thing happening. Even you could have a very important experimental results that could be interpreted as with a very little fold change, because it actually depends from gene to gene, it depends from expression to expression. Like as I mentioned in the previous cases, that if your developmental genes, they don't show a huge fold change differences, nevertheless their little fold change difference is very significant. You need to understand that what is the initial copy number of the genes in your sample. If the gene of interest or the gene what we are trying to interpret is a highly overexpressed genes, then a 10-fold change difference may not be that significant, because either way the gene is actually present in so many copies that even if a few copies are actually been altered, doesn't make a huge difference in the entire biology of the cell. However, if a few genes, say for instance, they are just present in a few copies, even a 5% change in their proportions is going to make a whole lot of differences in the entire biology of the cells. So we should not just go into what fraction or what, how much the fold change values are, but we need to go gene by gene, trying to categorize individually and study them as a separate entity, rather than have one common fold change and try to interpret everything in terms of that. This particular thing which is shown in the left hand side, this particular histogram, you could see over there the first experiment number A, that shows a lot of fold change, but look at the standard deviation, this is huge. That shows a lot of variations between those two experiments. That's not very reliable. We're not going to much interesting biology out of that. But experiment number 2 doesn't shows a huge fold change, but the standard deviation is very tight and it's less scattering, which shows that there's a very good reproducibility and very good repetition of the same informations even if you have performed several times. And that is actually very important. Rather than the absolute fold change, how much is the reproducibility, what it actually makes sense out of that small changes in the fold, even though it may not show dramatic changes in the fold changes. If you have seen the microarray data which is actually been represented in different journals or books, then you would see that vast majority of the time they are actually represented as log2 of fold change. So you could always question that why log2 transformations, why not just a linear scale? Well, I'm going to explain to you the reason in just very brief. Let's imagine the scenario as shown in the table, that there's an upregulation of a particular gene, say 2-fold, 4-fold, 8-fold, 16-fold, and then there's a downregulation, equal downregulations like 1/2-fold which is 0.5, or 1/4-fold which is 0.25, and 1/8-fold which is 0.125, and so forth. Now when you're going to see this particular data, then the upregulated genes are very nicely expressed and very nicely represented, 2, 4, 8, it's very easy for us to visualize it. But see about the downregulated genes, you could see that it's very hard to make a difference. It's very hard to interpret their actual downregulations, because once we go for the reciprocal of that, the value tends to get clustered and it's not very easy to feel the differences between these two. So we could always see that there is more upregulation and there is less downregulation. It kind of gives us that kind of feeling, even though it is an equal amount of downregulation. So in order to make it a rather more uniform, that the positive things which is upregulated are equally represented in an equally spaced way as compared to that of the downregulated genes, so we actually do a log2 fold transformations, and see on the right hand side what happens once we do a log2 fold change. So the 2 becomes 1, 4 becomes 2 on the upregulated one, but see the dramatic difference what you see once we do the log2 fold transformations of the downregulated genes. That actually shows real values like -1, -2, -3, -4, and so forth. So now it is very easy for us to interpret. So if there is an upregulation, 1-fold upregulation, or if there is a downregulation which is -1, and if there is a 2-fold upregulation, then a -2-fold downregulation. So you could see that now they looks very uniform, upregulated one and the downregulated one, and much easier to visualize and understand rather than having to deal with the fraction values. And that is the whole purpose why majority of the cases, most of the microarray data is actually represented as log2 fold change. And the lower panel actually show you a heat map, and that heat map is kind of once you have an upregulated genes and downregulated genes, usually you could have three different outputs of our microarray experiments. One is an upregulated fractions, another is the downregulated fractions, and the third is the fractions that really is neither upregulated nor downregulated, but in two different conditions. Say for instance, we have a drug treated and untreated control cells, and now you're going to ask that when you treat the drug, what are the pattern of the changes in the gene expression? Of course, not all the genes will be altered in the treated or the untreated fractions. You see a few of the genes are upregulated, few of them are downregulated, and the few of them remains completely unaltered. In fact, vast majority of them will be unaltered. Only small tiny fractions will be upregulated or downregulated. And all of this, whatever I am talking about, can be pictorially represented with the help of what you call as the heat map diagrams, where you kind of give a color code that if red is upregulation, then green is downregulation, and black is unregulated one, or there is no alterations in that, and that kind of gives you a visual representation of what's happening in your experiments right away without looking into the individual values. You could see a profile changing, and that is very interesting for us to interpret the data very quickly. So we are talking about the data analysis and the statistical considerations of the microarray experiments. So using statistical test is always a better way to determining the actual genes. That's what we are actually talking a little before. And once you do the statistics, one thing that actually is very important is the p-values to find the statistical significance. Like for instance, if you have two groups, say for instance a control group which didn't receive any drug, and a test group that is under the influence of a particular drug which you have added to that particular test cohorts, and then you need to find out that what is the effect of the change in the gene expression because of addition of this drug, and then whether this change is really significant, whether it is going to be really important for us. And for that we need to do what we call as the p-value testing or statistical testing. Now, in general, we want to maximize the sensitivity of our experiments, how many of them are truly changing without sacrificing the specificity. And this is we are going to talk a little now, that these changes whatever we see in the gene expression profiling, how true they are and how correct they are. Now let's talk now about the different challenges in interpreting the gene microarray data. Now the functional analysis of microarray data. Now the analysis at the level of the gene sets. So there are different pathways for doing the analysis. One of them is very well known, is known as the David pathway. Now what happens is in the David pathway is that suppose you have the control sets and you have the tests where you have stimulated with a particular drug, or you have the control cohorts and you have the cancer cells, so cohorts which has been derived from the cancer patients. We need to see that what are the gene expression profile changes between these two groups. Then once we have the differentially expressed genes between these two groups, we are going to apply this, we are going to take those differentially expressed genes and run them through a path which is called the David's pathway, which is going to take the entire gene list of the differentially expressed gene and put them into different functional categories. And that is very important to know that under which category the gene expression profile changes actually took place. So just telling an upregulation or downregulation is not going to be that useful, rather than just taking those entire data sets and taking those differentially expressed genes and putting them into different clusters. So we are going to create a pattern in the gene expression profile list, and that pattern is very important for us to understand what's the actual biology which is going on into this entire process. So the statistical significance, when you talk about, then we talk about something called the false positives and the false negatives, and you may be aware of the type 1 errors and the type 2 errors, and you may be also aware of the null hypothesis, where you're going to say that there is no difference between these two groups. So there is no difference in the gene expression profiling between the control cohorts and that of the test cohorts, and then we are going to either rule out or kill the null hypothesis, or we're going to follow that one, and for that we are going to do the experiments. The type 1 errors are the false positive errors, where there is actually a change and then we say that there is no change, and then there is something called the type 2 errors, which is kind of the false negative errors, where there is actually something which you are missing out, and that is actually something which is very important for us to understand. That if the sample sizes are large enough, one may have the statistical power to confirm the small changes. So you may not see every time a huge change in the gene expression profiles, and you might be surprised to see that even those small changes makes a lot of different interesting biology. So that's the reason why the statistical testing is very important, because it's going to find out the p-value and it's going to tell us that those small tiny changes still is very important for us to put forward the hypothesis. And we are talking about something called the multiple testing problems, where in an experiments like microarray, where we have so many gene expression profiling changes taking place in one single experiment. Now I'm going to give an example and tell you, like for instance, one of the most common thing that happens is the value of what you call as the p-value. Now you may have often see different manuscripts, they kind of put an p-value equal to 0.05 and then say that anything less than 0.05 is statistically significant, and then this changes, whatever we see is really the actual changes, the true changes. But I would say that that is an oversimplification in microarray, because of this reason that in microarray we are going to interrogate hundreds and thousands of genes together. I'm going to give an example to make you understand why that's an oversimplification. Say for instance, we take an p-value of 0.05, which actually mean that if you do an experiment 100 times, then 95 times it is going to be true, and then 5 times it is going to be false, either wrong, either upregulation or downregulation, whatever you see is going to be wrong. Now if you just do an experiment 100 times, so 5 times there is going to be an error. Now just think about, if you're going to do the experiment 1000 times, then 50 times you're going to make an error. And if you're going to do the experiment 10,000 times, then 500 times you're going to make an error. Now in microarray we are going to integrate hundreds and thousands of genes. So if we just allow that p-value of 0.05 to be a cutoff, then we are going to carry with us a large number of false positives or false negatives. That might be okay if you are going to do with a small sample size, but for an big experiments like microarray or mass spectrometry, where you are going to integrate hundreds and thousands of profiles in one experiment, it's not going to work, because we are going to carry with us a large number of false positives or false negatives. And in order to rule away with that problem, there is something called the FDR corrections, which is false discovery rates. It is kind of something like that okay, you have this p-value, and then you are going to apply additional algorithms to make this p-value even more stringent, so that we don't take with us too many false positives or too many false negatives. And that is what you call as an FDR corrections. Now what the FDR does is that it actually makes the p-value a little more stringent, and so that we don't carry with us large number of false positives. I understand that at this point of time you know what false positive is. Something, for instance, we know that hypertension is actually associated with cardiovascular disease. Now but everyone with a hypertension, with a high BP, doesn't have a cardiovascular disease. So just measuring high BP and then telling that okay, this individual is going to have the cardiovascular disease is something like in false positives, because not every cases is going to have the real disease. So in that, this similar kind of scenario happens over here, that if we have a few data sets, and if you go by this 0.05 FDR, 0.05 p values, then we need to further make it more stringent, so that we don't carry with us a large number of false positives, which becomes a real problem in this kind of assays where you have hundreds and thousands of data points. And that is what we call as the false discovery rate correction, and that is very important in this kind of experiments. The next what I was talking about is that once you have the data sets, how we are going to cluster them into different functional categories, so that you can find out a pattern within this data. And there are different ways of clustering. Again, this is not the module which is going to talk a length about this clustering programs or the different clusters, but just remember that majority of the clustering programs actually works on finding out the distance between the two data points, and try to see that which distances are very minimum, which distances are maximum, and then they are going to cluster them together into different patterns, so that we know that sets of the genes which belongs to a particular pathway stays together. So there are different types of clusterings, and again someone is going to possibly talk on the different clustering algorithms. We are not going to go into this particular module in too detail. So once we give microarray data, then the next important thing is that what are we going to do with the data. So the thing is that we are going to see the pattern in the data. We are going to look for the patterns in the data, and that pattern is very important to find out what's really happening in the experiments. So there are different ways to find out the pattern. One of the ways what you call as a clustering of the entire data sets into functional clusters, so that we know that these two clusters are similar and these two clusters are different. There are different ways of doing the clustering, and the basic fundamental maths that goes behind this clustering is finding out the distance between these two data points. And if the two distances are very small, these two data points lies very close to each other, so they're related to each other. However, if you find that there's a huge distance between these two data points, this makes us to assume that they're possibly of the different clusters, and therefore they are spaced out from each other. in the three-dimensional space. And therefore this is very important for us to take the entire data and look into the patterns within this particular data. This is the matrix of the gene expression. We are just talking a little while that we are going to find out. What we're doing is that we are actually measuring the distances between these two data points and then trying to make a sense out of it, that which distance is the maximum and which distance is the minimum. And if the minimum distances are there, that actually means that these two genes actually behaves very similar, or their pattern actually changes together. And if they are very far away from each other, this means these two expression values are very different, so they belong to the different clusters. So that's a heat map, and you could see how nicely the genes which are actually been there is been clustered into different categories, and this is what we call as an hierarchical clustering. Over in this one, we could see that give a particular color codes associated with a particular process. Say for instance, if this is up regulated gene, then you give them a red color, and if it is in down regulated gene, you give them a green color, and trying to find out that exactly how the genes are actually related with each other. So at the beginning, each gene is a cluster, and in each subsequent steps the two closest clusters actually been merged until one cluster remains. There are a few different ways of doing that. This is what has actually been depicted in this particular image. So we are talking just now about the clustering, and K clustering is one of the way, and then there is something called the hierarchical clustering, which is another way of, just these are the different ways of taking the entire data sets and then dividing into different functional categories so that we can find out the paired pattern within this data. And that is all based upon the finding of the calculating the distances between the two data points. So that is very important, and most majority of the time we do this, and then we create what you call as a heat map. So heat map is going to give you a visual interpretation of the data, and then is going to tell us the sets of the genes which are upregulated and the sets of the genes which are downregulated, and the sets which really doesn't changes between a control or cohorts or the test cohort. So that's a very good visualization tools to just look into a glance into the entire data and kind of reaching to a conclusion what's happening with your data sets following a particular experiments. So students, coming to the end, so let's now summarize what you have learned in this module. Is that gene expression profiling is very important, because what is at the end the functional output of the genome is the mRNAs or the transcription changes, and that is what is actually being communicated in the form of the protein changes, because we know that transcripts go and translates into proteins, functional proteins. So microarray based gene expression profiling is one of the very common tools which is actually been in practice for the last several decades to find out the change in the gene expression profiling. Having said that, that's one of the tool. Of course, sequencing based technologies are picking up very fast, but nevertheless still microarray is the tool of choice because of several advantages which I just mentioned, like they are much more cheaper to perform, and they doesn't need that rigorous statistical tools, and at the same time they are equally informative. Of course, there are certain pitfalls of that which we are not going to talk in this particular module, but it is going to be very important, at least for a particular lab, to get a hold on some of the pilot data out of this particular microarray based techniques. And what we are trying to find out in microarray is the differentially regulated genes between these two groups, the control cohorts and the test cohorts, and then take these differentially expressed genes and then trying to find out, put them into different algorithms, clustering them into different categories, and then running them through the different pathways so that we know what are the different ways they actually forms a pattern, and individual patterns, they belongs to what kind of pathways, genes belonging to what kind of networks, and then we can actually build an entire pathway or network out of that, and that is going to be much more informative to address the biological questions. So be aware that microarray based technology is still the method of choice, and all of you can actually pick up a few of these microarray based published data or papers which discusses about this, and you will see that there are a lot of interesting interpretation which we can reach from just performing a few microarray experiments, and that is going to address a lot of interesting biological questions which we are trying to address. Thank you.